Well,
Go right ahead.
It is my pleasure to introduce Carol Malice, the Executive Director of Rhode Island Bio, Rhode Island's Life Sciences Trade Organization. We at District Hall, Providence, are very excited to partner with Carol and Rhode Island Bio to launch their new Women in Science series. This series highlights the accomplishments of women in science and provides an opportunity to learn more about their personal and professional journeys and how they've overcome challenges along the way to achieve success. So without further ado, take it away, Carol.
Thank you, Tuni. Always great to partner with you and the team at your place. So thank you so much. Good morning, everyone, and welcome to the launch of our Women in Science series. As Tuni mentioned, I'm the Executive Director of Rhode Island Bio, which is Rhode Island's Life Sciences Trade Organization. And what we do is provide our members with information sharing, advisory services, resources, training, and everything they need to be successful in the life sciences. So part of that is looking particularly at women, and we've had a lot of interest in helping women in the sciences. So we decided to launch this series, and it's designed to provide a forum and supportive environment to share best practices, provide insight, and offer advice on leadership and professional development. So before we start, I want to thank all the members of our planning team who helped us launch this. We've been working for many months on getting this started, and that includes Alyssa Boss, a Rhode Island Bio Board Director and partner at Cameron and Middleman, Sarah Bramblett from the Department of Labor and Training, Olivia Byron from the Department of Labor and Training, Stacy Carter, our Rhode Island Bio Program Director, Brianne Meehan, HR Director at Rubius Therapeutics, Patrice Milos, our board chair and vice president of scientific operations at Pine Trees Health. Katie Sharkey, Dr. Sharkey is the associate professor and assistant dean for women in medicine and science at the Warren Alpert School of Medicine at Brown University. Melissa Simon is the director of business development at Brown Technology Innovations, and Vera Tiagi is the director of human resources at Amgen. So thank you all for your great work in getting this up and running. Just a few logistics before we get started. We are going to ask you to put your questions in the chat function, and then we will take a look at those, and Katie Sharkey will be managing that function, and we'll have time for Q&A at the end as well. So now it gives me great pleasure to introduce today's guest moderator, Dr. Katie Sharkey. She is the Associate Professor of Medicine and Psychiatry and Human Behavior at the Warren Alpert Medical School of Brown University. Dr. Sharkey is board certified in sleep medicine, internal medicine, and psychiatry. She's the medical director of the Sleep for Science Research Laboratory of Brown University and is a fellow of the American Academy of Sleep Medicine. And she's also a great advocate for women. So she has done a lot at Brown in her life on behalf of women, and we are so proud to have her on our planning team. So take it away, Dr. Sharkey.
Thanks, Carol. Thank you for that. And I'm so honored to be on this committee. It is when we first started having this conversation about getting this series up and running in the fall, to have made it to today just feels so exciting. And I'm really thrilled to bring so many people under the big tent to have these conversations about how to advance women's careers and how to make science an attractive professional option for women. And I couldn't be more delighted that my colleague and role model and someone I look up to so much, Dr. Phyllis Dennery, who I will tell you about in a minute, is our kickoff and inaugural speaker for this, because I can't think of anyone again, who I look to in meetings, in day-to-day, grand rounds, and look and say, what's Phyllis's bearing? She really is somebody I watch closely and want to emulate. So to get a chance to hear her talk about her career, I'm terribly excited. I also want to acknowledge it's been a hard week for a lot of women, and that we're probably all doing a little compartmentalizing around being able to bring our best selves to this meeting today. But in the spirit of staying hopeful about our careers and all wanting to be like Phyllis someday, I'm glad we're getting to spend this hour together. Let me tell you about our esteemed speaker. So Phyllis Dennery Dr. Phyllis Dennery is the Sylvia K. Hassenfeld Chair of Pediatrics at the Warren Alpert Medical School at Brown. And she's also the pediatrician in chief at Hasbro Hospital, and holds, in addition to those pediatric professorial appointments, a basic science professorial appointment in molecular biology, cell biology, and biochemistry at Brown. And so in terms of someone who bridges the Rhode Island bio life sciences community between sort of the very clinical day-to-day hands-on patient care and, pipetting, she does it all. And that's one of the reasons we thought she'd just be perfect. I know she's going to tell a lot of her story, but I'll just give a quick rundown that she did her bachelor's degree, at McGill, followed by her medical school at Howard, and then trained for pediatrics at Children's National in Washington, D.C., and completed her fellowship in neonatal and perinatal medicine at Rainbow Babies which is part of Case Western in Cleveland. She has so many accolades and is recognized nationally and internationally as a leader and an expert. I'll just highlight a couple of her accomplishments. So she serves on the Secretary of Health and Human Services Advisory Committee on Infant Mortality, and was elected in 2014 to one of science's highest honors, the National Academy of Medicine. And I, Phyllis, when we first got on, we were fooling around with the Zoom a little bit. And so I will share briefly, so Phyllis shows up for people. She is a person who's got your back, who's supporting others, who's like lifting her colleagues and the trainees and junior faculty for her. Last night though, she showed up in my dream. So Phyllis, I dreamt that we were doing this whole thing from my cell phone, and I had to try to prop it up. So whatever issue you have with Zoom is not going to be as bad as it was in my dream last night. I'm not kidding. I'll tell you the whole thing later. It was crazy.
Don't count on
But
me.
so with that, I just want us to warmly welcome Dr. Dennery, and we're so glad you're going to share your story so that we can all try to grow up and be like you. Okay,
Thank you.
take it away.
Katie, this is way too much. And I appreciate the positive remarks, and I aspire to be what you said I am. I'm going to do my best to share my screen. I will give an advance notice that my cell phone is my internet connection right now because of technical challenges at the university or here at the hospital, but let's give it a whirl. So here today, when I asked Katie, what would people want to hear if I'm giving a talk? Because, it could be a variety of things, a mentoring talk, a sort of career without science talk. And she insisted that I put in the science. So I apologize in advance to anyone who is bored by the science. So I will start by saying, first of all, I have nothing to disclose, but I make the analogy of trees and winding roads in this talk, because trees are, to me, the representation of how research ought to be, with branching, this is the other thing, branching, opportunities along the way. And oftentimes in science, we go along a winding road and make changes along the way. So that's kind of the message. For me, it all started with having a real passion for the field that I specialized in, neonatology. I was fascinated by these tiny little people, as shown here, a tiny person who could fit in the palm of your hand or even smaller, who needed support and help. And one of my biggest mentors then was Maria Delavoria Papadopoulos, who just passed away last year. She was the mother of neonatology and did some very pioneering things around trying to help keep babies alive. So neonatology is not that old of a science. And I always like to come to a little history because it's kind of fun to talk about the fact that back in the early 1900s, we thought of neonates and prematures as little freaks, essentially. They were put in these little um, trash shows and were cared for by these wonderful nurses who did all of the work. And of the 8,000 babies who were taken in these incubator shows, 6,500 survived, which is quite remarkable. And I'll show you here, this is what would happen. People would pay their quarter or whatever, and they would peer in to see these tiny little babies in these incubators. And we've come a long way from then, but it's quite remarkable to think that there are still survivors of this era who actually did a nice special on NPR a few months ago. So if you ever get a chance to hear about the incubator babies, there are people, the survivors, who are talking about their experience there. So why do I care about these babies being small, et cetera? It's because these babies have a disrupted development. So they have been in an environment that's conducive to growth in the mother's womb. But this thing is going to do this, but they're born, all of this wonderful normal development is interrupted, and they are allowed to come into the world, and they have immaturity of the brain, the gut, the kidneys, every organ, and also at the same time are under stress, are not getting adequate nutrition, and don't have good placental transfer of nutrients that are necessary for their growth. I was trying to explain this to when I was interviewed in a program in Philadelphia on national television, which was most embarrassing because I told the lady, I said, well, it's like baking bread. I know some of you have adopted baking bread. I have during the pandemic. It's really fun. But you make something, a dough, and if you cook it and it's not quite ready, it's never going to be exactly right. You can't do much to correct that. And it won't be like those perfect loaves that you would have achieved if the full baking process would have taken place. That's a little bit like prematurity. It's not being small, it's being unable and immature and unprepared. So that's the same story. They're a little bit half-baked. But let's look at the lung a little bit more seriously and see that, in the lung, there is alveolar development going on in the first, as the baby progresses towards maturity. So at 38, 40 weeks, the lung has made these alveoli that are important for gas exchange. But before that, you don't really have those structures. And babies are born at maybe 34 weeks, like JFK's son, who was born then and died. Oh God, this is annoying. Sorry. Who died prematurely of this lung disease because his lungs were underdeveloped. Now we can resuscitate babies that are maybe 22, 23 weeks in gestation, and their lungs are extremely, extremely immature. So that question intrigued me a lot, and I wanted to better understand it and also understand why lung maturation, how could we change things to better define lung maturation. If you notice here, the younger you are, the more likely you are to have lung injury in response to any stimulus that occurs after birth. So the odds of injury double with each decreasing gestational age. And the condition that these babies end up with is called bronchopulmonary dysplasia. And that was sort of the focus of what I wanted to do in my career. And what gives bronchopulmonary dysplasia? All the things we do in the nursery. We ventilate these babies, we give them a lot of oxygen, perhaps. We do all sorts of things. They get inflammation, infection. That may cause it. And as a consequence, the lungs look abnormal. So if you see here, this is a normal CT scan of a lung. It looks pretty boring. It's just black, not much going on except a few vascular structures. Look at in contrast, what happens to a baby who has severe BPD, that lung disease, and you can see a lot of these sacs of fibrous strands and little holes that are more like a person with a chronic obstructive pulmonary disease, something that Katie knows a lot about. And then as they age, these things don't disappear. They don't return back to normal all the way. There's still these opacities. And in one recent study, they've shown that these exist even in adults who had BPD as babies. And this is a huge public health problem because it affects a lot of the babies. There's 7,000 new cases a year, high morbidity, rehospitalization, lifelong alteration in lung function if they get this disease, and increased chance that they will have neurodevelopmental delay, other delays. So we can't focus on every last thing that impacts BPD, but we were very interested in oxygen, because oxygen has a role to play in this. Too much oxygen can inhibit the growth of the lung and make for a lung that has BPD. So why is that relevant? Well, when a baby is inside the womb, it's like being on Mount Everest. Clyde Wright, who is a colleague of mine from Children's Hospital of Philadelphia, would often quote Sir Joseph Bancroft. On Mount Everest in the uterus is what the baby normally should be in, where the oxygen concentrations are very low. When the baby transitions to air breathing, just breathing regular air, you can see that oxygen concentrations are a little higher, triple. And this is Clyde here. And at the highest level of oxygen, when we deliver extraordinary amounts of oxygen, 100%, then that oxygen concentration is driven up to very, very high levels, which can be toxic. We've been using oxygen for many years. This is a contraption that they used to use in the early 1900s to deliver oxygen to this little baby in here, because some of the babies that are premature tend to stop their breathing and they turn blue. And so rather than do something manually every time, they would place them in these chambers and give them oxygen. We still use oxygen either through a ventilator, what's called CPAP, which is pushing air into the lungs, giving just a nasal cannula. And I would argue that in the smallest babies that are preterm, for them, just being in room air is too much oxygen probably. And we've tested in animal models to show that oxygen can cause an arrest in lung development, just like it would in a baby who has severe BPD. Not just a ventilator, but just oxygen in and of itself can distort the lung. So shown here is a baby with severe BPD who died, and here is the lung of a mouse exposed to high oxygen.
I'm listening.
Someone is talking. And so this lung is distorted, as you see here. Okay, so that's sort of the premise of what went on. And I went with that interest on to my fellowship at Case Western Reserve. And I went for these two guys here, Doctors Averoy Faneroff and Richard Martin. These two individuals had written the book on neonatology, and there was a lot of lung research going on, but mostly control of breathing. I was interested in understanding how oxygen could cause reactions in the lung, forming oxygen radicals, which would then go on to damage the lung in the presence of iron and other substrates, causing oxidative damage and impaired lung function and lung death. And so, I was also interested in the fact that the body was smart enough to generate these antioxidant defenses that could help protect against these radicals. So that's how I started my research career. And I had a passion for understanding how the lung defended itself against oxidative stress that could lead to a cascade of reactions here and also lead to a very abnormal looking lung. So I did some work in my fellowship, and that gave me a chance to be competitive for my first faculty position at Stanford University. I show David Stevenson here. I'll show lots of pictures of people because to me, these are the people that help inspire you and help you get to where you need to go in your career. So other than the beautiful sunshine and everything else, I chose Stanford for the fact that there were mentors there who could help me and help me better understand how to move forward in my research career. So the first hurdle was to establish a niche. Um, these individuals, David Stevenson and his colleagues, were very interested in the brain and jaundice. If you notice the jaundice brain here, and what it did to babies, where after severe jaundice, you might end up with cerebral palsy, with what the consequences of what's called kernicterus. And I kind of wanted to understand, well, I had to figure out how am I going to blend my interest in the lung in the context of an environment where they're mostly interested in bilirubin. One of the gastroenterologists approached me as I was leaving my fellowship at Case Western and said, have you ever thought of bilirubin as an antioxidant? I was interested in antioxidants protecting against oxygen radicals. So how could I utilize this to better understand something that would be of interest to me and create a niche? So how could I incorporate my interest in oxidative stress and the lung with the knowledge about bilirubin that existed there? Well, these individuals came in very handy. Dr. Anthony McDonough was at UCSF, which was about 45 minutes up the road. And he inspired me because he had the availability of understanding the role of jaundice because he had a colony of gun rats, which are rats that are jaundiced, and understand how they might impact the lung, their lungs and oxidative stress. So look at the reaction. So bilirubin is an important reaction in, and its catalytic enzyme is hemoxygenase, which I'll talk a bit about later. And here's Hank Freeman, who was very much entrenched in David Stevenson's lab and would help me get these experiments going. Doug Spitz was at WashU. I met him at a couple of meetings and we started talking about things, and he helped me with some of the assays that I had to do. And Rod Levine was another person I met at these meetings who was at the NIH and provided me a lot of counsel as well. So you need a lot of people is the message overall. And Camille Faulkner was the first undergraduate student in my lab. She was a young lady who wanted to understand more about science. And she and I worked together and bumbled along for those first years to try to get to the point where we took these gun rats that you can tell that they're pretty yellow, and their litter mates, which are not yellow, and exposed them to high oxygen, hyperoxia, and then looked at the relationship between oxygen radicals and serum bilirubin. And we uncovered that the lower the bilirubin, the less protected they were against these lipid peroxides, these hot oxygen radicals. So that was a very important discovery for a starting professor, assistant professor, and I show the tree to say that this is the beginning of my little research efforts at that point in time. But more important was the gene that regulated bilirubin production, and that was hemoxygenase. Hemoxygenase is highly responsive to oxidative stress. So I thought, well, maybe let's focus on studying hemoxygenase and what it does in the newborn lungs in response to hyperoxia. So when I went to Stanford, one colleague, Chris Kontag, that I will show in a bit, had developed this technology based on light passing through tissues and the generation of light by luciferase, luciferin, which is found in the firefly, and how that would react with a construct of some sort where luciferase was expressed in the presence of luciferin to generate light. Of course, you needed a live system for that, and using a nifty light in vivo imaging system where an animal was placed in a chamber, and after being constructed with these special light-driven promoters, and here's Chris showing off a little bit with his fireflies, but you could capture this on a computer and look at light. So that was some of the work we started to do, where we compared adult mice with this hemoxygenase promoter after exposure to hyperoxia. You can see their lungs are lighting up, compared to the newborn, where here's the newborn. One was expressing very high levels and the other one was less so. But what happens over the course of the hyperoxic exposure is they have less hemoxygenase in their lungs. So that was an interesting discovery that we continued to work on. And I had Michelle Shagog, who's now an ENT doc, or I'm sorry, an orthopedic doc at Guangyang, who is one of the most incredible people I've worked with, who was in my lab first as a postdoc, all the way to lab manager and director of the lab, until she didn't follow me along after a little while, and Sasha Bratnova. And we conducted these experiments to better understand what happened with hemoxygenase. So you need people, you need trainees, you can help people get excited about science along the way. So that was another little tree growing in that direction. We ran into this wonderful young lady who said right away when she joined my lab, she was a postdoc, she was a neonatal fellow. She said, I want to have a lab experience, but I'm not interested in ever having a lab myself. I want to be a clinician. And she's now the big director at San Diego Children's Hospital. But she was a mathematician, a precise person, and she wanted to have a project that had meaning. So I said, well, why don't we try to overexpress hemoxygenase and see what happens to cells to see if they make it better or worse. But what happened is, instead of seeing hemoxygenase expressed where it normally was expressed around the cell, it started going inside the nucleus, which was not where it was supposed to go. And Denise observed that, along with another wonderful man, Yihao Wang, who was from Taiwan. He had come to spend a year or two. And when he first approached me, he truly had we could only communicate by writing things out, because it was very difficult and challenging for him to be in the laboratory and understand, yet he did tremendous work. And this other young man who wrote me from Germany and said, I want to spend some time in your lab. He is now a specialist in internal medicine. So he joined the lab as well and started working in this area. And so more's going on. But what happens? Well, occasionally decisions are made to go somewhere else. And I had a great opportunity to run the division of neonatology at Children's Hospital of Philadelphia. So I kind of said, well, I'm going to do that, and ensured that I would have sufficient resources in the laboratory to continue to work. And most importantly, I said, please, Guang, please come with me. Leave California, hard ask, and come to Philadelphia to help me establish myself in the laboratory. At that time, as we talked about, we were starting to be intrigued by what would hemoxygenase do in the nucleus, inside the nucleus, and said, let's try to understand how that happens. So we know that hemoxygenase was anchored to the smooth endoplasmic reticulum, and it was a hydrophobic molecule. So how could it enter the nucleus? So Sebastian, strangely enough, he chose to come and spend some time in Philadelphia during his residency. I wish American systems allowed for that sort of thing. And Sarah Lynn, who's now at the NIH, she's a big director there, she joined the lab, and they figured out that hemoxygenase was cleaved at the C terminus and could enter the nucleus through this cleavage process, and that things like hyperoxia accelerated this cleavage. That was a big finding and an important one for us in the lab as we came to a new environment. And more work was done to dissect out these different forms of hemoxynase from the full length with its C terminus to a form that had a truncation of this C terminus to a form that also had the truncation but eliminated the enzymatic site, to conclude that hemoxynase could serve as a signaling molecule, not just an enzyme that degraded bilirubin. And others joined the lab, such as Ping La and Andrew Parsons, who's now a medical student at Harvard, and Patrick Fernando. So this work was more to say, okay, this is a niche that I'm developing, I'm characterizing this protein, and so forth. And also at the same time, I had the opportunity to go to Japan and give talks. And Fumihiko Namba, Fumi came to the lab for 2 years and decided to expand our findings by developing these mice that overexpress the different types of hemoxygenase, the nuclear form, a form that expressed in the cytoplasm at lower levels, and a form that expressed at very high levels in the cytoplasm. Sometimes you find things that you didn't expect. The very high expressing form of hemoxynase was associated, when we did these scans of the lung, with lung lesions, like proliferative lesions. So is that a cancerous lung? We worked on trying to better understand that, but that was a very complex understanding. Meanwhile, Guang was working hard on trying to define what hemoxynase did exactly in the lung, which was a task in and of itself. And it looked like it caused some simplification of the lung. She developed this model where we exposed the mice to hyperoxia for 3 days and then allowed them to recover. And the injury was most prominent in the recovery, not in the initial injury, which was an important finding. Further work was done to show that when you disrupted hemoxynase, you completely dysregulated lung gene expression. And that work showed that you upregulated cell cycle genes, transcription factors, and also downregulated other transcription factors and cell cycle genes. Still a little confusing and a little unclear as to what is hemoxynase doing exactly in the lung in response to hyperoxia. So after 12 years at University of Pennsylvania, I got the opportunity to come to Brown. I didn't know much about Rhode Island. I didn't understand what I was doing, but I thought, oh, I'd like to be a chair of a department of pediatrics. And, you know, in a crazy way, oftentimes you are brought to the attention of people, not so much because you're a brilliant managerial person, a strong person with budgetary skills, but rather based on your science. It's kind of a little counterintuitive, because does that mean you'll be a great department chair? It's not clear, but someone thought I might be good at this, and I ended up here at Brown. I was intrigued by Rhode Island and the fact that there was a lot of things that could be done in a community that was so tight knit, and also hope to continue my research. Well, I hope to continue my research, and it was very challenging to do so in the beginning. And my tree was dying a little bit because I had to rebuild a whole lab. I tried to convince Guang to move with me, to help me get started. She came for a month and just said, look, my family's in California. I came to Philadelphia for you. I can't keep going further and further away from home. My kid's going to college. To high school, I have to be there for my family. And I totally understood. So I was very worried. I had to rebuild a laboratory from almost scratch. There were projects that were still ongoing towards the end of my departure at Children's Hospital of Philadelphia, such as this connection between hemoxygenase through its regulation of heme and the circadian genes and circadian gene expression. And I had the greatest opportunity to train, or he trained me, this young man who came from, to me as a first-year undergrad, very green in terms of research, but very interested. And he, with some of his reading, some of the experiments that were done, identified this protein, reverb alpha, as something that was regulated by heme oxygenase. So when we looked at our construct with this reverb alpha protein, which is part of the circadian rhythm, and treated the cells that were overexpressing this with a siRNA, so a silencing RNA for HO1, we lost the signal for the reverb alpha. And Maurice, an undergrad, a person who was waiting to go to medical school, was the one who did the bulk of this work, and he won awards that were normally given to fellows. They didn't recognize that he was only an undergrad. So I'm so proud of Maurice. He's now a physician, internal medicine specialist in New York. Shaun Sengupta joined the crowd looking at this reverb alpha, and we collaborated with Lauren Francie here and others in Cincinnati to show that in a circadian oscillation model through a machine called the LumiCycle, we were able to suppress the circadian oscillations just by expressing a construct that had heme oxygenase, that reduced heme oxygenase. So heme oxygenase seems to be very important to circadian biology, but it was hard to continue this work once it came to wherever I am now, to Hasbro Children's Hospital and Rhode Island, because I had to rebuild the lab, find the people, start back up, try to get people trained. We were fortunate enough to meet Monique de Pape over at Women and Infants, who had samples from the lungs of premature babies born at 25, 23 weeks, and also who had received oxygen and ventilation. And these young babies, we were able to show that heme oxygenase was highly expressed in these lungs. Well, so yes, so how do we go from there? Well, at that point in time, I hired a few people in the lab who had other types of expertise. And rather than beat my head against the wall, I put that work on pause for a bit and took a little turn in the road. So we looked at, we were interested in hyperoxia, that interest never stopped, but how could we leverage some of the interests that existed around metabolism, and how could that be something we could learn about? Mitochondria are very important to regulate metabolism. Oxidative phosphorylation and glycolysis are very key functions in metabolism. And there were nifty tools available that allowed us to measure cellular metabolism by injecting a variety of drugs and so forth into the cells that are found here, and monitoring the response to these drugs that are metabolic regulators. So this allowed us to measure both mitochondrial respiration through a variety of drug injections, or glycolytic function for glycolysis. I won't go into any of that detail. And, when in Rome, do what you're supposed to do. So we tried to understand mitochondrial fuel utilization, in the context of hyperoxia. And also we went back to our heme oxygenase model to understand what changed in these mice, the knockouts versus the wild type, in response to just hyperoxia, and also just at baseline. And this was published recently by Hongwei Yao, who is a senior scientist in the lab. He came from Rochester, joined the lab, and he's been nothing but a machine in terms of getting work done. And I've greatly appreciated having him there, as I did having Jen Carr, who was also familiar with Brown, and she worked on projects relating to mice with hemoxygenase and their insulin sensitivity and other metabolic findings. So we were able to get back a little bit on track, but we got we started paying more attention, sorry for my computer clicking back and forth, about altered metabolism and how that might influence cells. So cells that are highly proliferative often utilize glycolysis, and that's referred to as the Warburg effect in cancer, compared to normally well-differentiated tissue, which tend to utilize oxidative phosphorylation more for a source of energy. So we wanted to look at that in our model of hyperoxia. So we took mice, exposed them to hyperoxia for 3 days, and then allowed them to recover for prolonged periods of time in room air. And here's Abigail Peterson, who was the lab manager. She knew mice better than she knew humans. She was able to collect lungs and do all this work to try to understand what happened to metabolism in the context of hyperoxia. So we showed that mice exposed to hyperoxia had reduced lung glycolysis when exposed to hyperoxia for 3 days, and that persisted even up to 60 days. So that means that you change the metabolism of the lung with even an early insult. And we tried to understand how that happened, and P53 became a focus of interest. And we met this wonderful young man, Alejandro Scaffa, who's now at Pfizer, who spent 3 years actually as a doctoral fellow as a doctoral student in the lab. And he used various technologies to create knockouts of P53 using CRISPR technology, which I won't go into detail, and showed that when you changed P53, you could change metabolism in these cells in response to hyperoxia. So you increased cell metabolism, meaning that P53 was an important mediator of reducing cell metabolism in response to hyperoxia. Oh, I'm so sorry. So hyperoxia can also regulates P53, but that pathway to glycolysis, there's also a pathway towards what's called senescence, so aging. So interestingly, there's a huge aging focus at Brown. There's a Center for Aging, and Abigail was a member of a lab that was very much focused on aging. He says, well, why don't we look at senescence in response to hyperoxia? Well, sure, why not? So we started to look at that, and senescence is an important state that prevents the cells from proliferating. And it's an important tool against cancer, against wound healing, but it's also very important in aging. And there were techniques to understand senescence that we developed in the lab and utilized. And bottom line is hyperoxia in a cell line, a lung cell line, caused these aggregations that were consistent with senescence. So hyperoxia was able to cause senescence. And when we suppressed P53, we were able to see a difference in senescence, a reduction in that senescence. So P53 increased glycolysis, but reduced senescence. So how are we going to know which cells in the lung were doing this? So we looked at a staining technique called lamin B exclusion. So all the cells that have that little yellow donut are normal. The cells that don't, that are blue, are senescent. And we noticed that in hyperoxia cells, there were more senescent cells. And we looked over the course of our exposure to hyperoxia, and only at day 7, postnatal day 7, did we see this peak in senescence, albeit very small quantities. And we tried to understand, well, what cells in the lung are becoming senescent? Wouldn't that be important? Are they lung cells? Are they capillary cells? Are they neutrophils? What cells are becoming senescent? The lung is such a mess, so many cell types. And each of the cells has an important function. We have cells that are producing surfactants, cells that are lining the lung and allowing for oxygen to diffuse, and other types of cells. So how are we going to find that? Well, we started with just using different staining techniques. Oh, was it this kind of cell, the epithelial cells? No. Was it another type of cell? Well, we did a lot of work. Abigail did a tremendous amount of work to understand which cells were in fact senescent. And the answer was she couldn't really tell very much. So we had to think of something better. And this is where the story ends soon in this saga. We partnered with some people who, computational biologists, that was through Alejandro's connection. He said, why don't we do single cell RNA-seq? We would be able to dissect the whole lung into a single cell lung suspension, barcode these cells, do next generation sequencing, analyze these data, and understand what each cell was contributing to the process. Well, that's not my area of expertise, but I thought, sure, that sounds fun. So we were able, with the help of Nathalie Hulin, to dissect out all the different cells in the lung. Look, it's not a uniform population. There's so many different cells in the lung, and how those cells changed in response to hyperoxia. And that was characterized by these Tisney plots where you can see differences between air, hyperoxia early on, hair, hyperoxia later on. It's not as important to know the detail of this, other than to say that with that, we were then able to say, well, what is happening specifically in each of these small little clusters of cells that may be type 2 cells, type 1 cells, but are unique? And how can we better understand each of these cells? We realized that they were losing some of the progenitor cells with exposure to hyperoxia, and that stayed permanently. And also that we were gaining other types of cells, which were very interesting because these cells express a long non-coding RNA, which is important in cancer. So is it possible that by hyperoxia, I apologize, prolonged short hyperoxic exposure and recovery, we are creating more cells that have a potential for becoming cancerous? That's the ultimate question that we're finalizing in this paper that we hope to send out very soon. We will also use this technology to isolate out the senescent cells and understand their characteristics. Which cells are senescent? Which cells are causing a problem? Are they disappearing? Are they staying in the lung? Those are all things we can continue to do. And continuing the connection with our Japanese colleagues, they're interested in silencing microRNAs. And are these important in hyperoxia? And can they be a way to suppress or accelerate senescence? This is Hajime Maeda, who joined us in the fall, in the middle of a pandemic, in March, and has had to adapt to life in America without much but isolation, which is really challenging. So as a consequence, I apologize for all this flipping, as a consequence of all of these things, we're left with thinking about the fact that a high oxygen that affects our babies can change the lung radically, can change the types of cells in the lung, can lead to excessive proliferation of some cell clusters that might lead to cancer, may alter the lungs' function, and I didn't even get into conversations about pulmonary hypertension or endo-MT that Hung Wei is focused on. But so many things can happen as a result of this hyperoxia that occurs early on that leads to long-term consequences that may drive the lung to be very different as a result, and may explain some of the injury that I was most concerned about when I was embarking upon this journey. So what are some of the quick lessons learned? Well, along the way, I told you my interest was to do no harm, to care for these babies, to do what I could to make them better. And that is still the goal. That's still what drives this research and what drives me to be excited about the work that we're doing. I think along the way, I've also showed you that it was very, very important to reinvent myself at different points, to change, to adapt to the environment, to not stay in despair, but to break the barriers and to continue to change and alter as the world presented itself. I will point to serendipity as an extremely important concept in all of this. Sometimes you have to go with the opportunity that presents itself, and it may lead to a happy or beneficial result. And I feel that that's happened several points in time along the way, serendipitous discoveries. I will also talk about pandemic productivity. There's that crazy silver lining. While we were on lockdown starting in March, the team led by Hung Wei, who was dynamically excited about getting things out, looked at a lot of the written data, the data that had accumulated that wasn't transformed into a paper, and started pulling things together. We were able to throw out 8 publications in from the time of the pandemic, which was more than we had done any year before in recent years, because there was downtime to think, to regroup, to take advantage, to reroute. Oh dear, what happened? Can you still see my screen? I hope so. Then with all that, your tree can keep blossoming and growing, and you can take advantage of the, 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 the bumps in the road, the turns in the road. I think none of this can happen without mentorship. And I told you about some of these mentors along the way. Why do I have Condoleezza Rice? I'm not a Republican, I'll say that right up front. But she was at Stanford as the provost, and she told me, you know, Black women often are thrown into opportunities that wouldn't be given to others. And oftentimes that may short circuit some of the work you're doing as you take on more responsibility. And she was extremely correct about that. As I have left Penn, I've not left my mentor, Dr. Arthur Rubenstein, who has guided me along the way and for leadership decisions and other words. And some of you may obviously recognize Jack Elias, Christina Paxson, and Rick Locke, who serve as advisors here, both leadership-wise and otherwise. I want to acknowledge all of the collaborators along the way. There are many and too many to name by name, but all the labs where I had an opportunity to do some of the work that I've done. And the lab here at Brown with the new set of people that have come and joined along the way, and the Brown Computational Biology Group, which has helped in this latest endeavor. We were blessed with funding as well along the way, although that came and went, unfortunately, and came back. So thank you to all the agencies that funded the work. And I can't forget Monique DePape, who was kind of a bridge and continues to provide us samples so we can make relevant findings to humans. So with that, I thank you for your time, and I look forward to any questions.
That was a tour de force, and it was such a pleasure, Phyllis, to have you put that together for us and sort of take us down the winding road of where one discovery led to another. And, you know, there's so many themes that I'm going to be thinking about for days to come about this talk, how as advances in technology changed, that gave you a chance to sort of chase down a different question that 2 years earlier you didn't have the ability to do. all the faces that came in and out of the lab, and I'm sure how they inspired the different ways that your tree grew. I'm going to read out, so we have time for a couple of questions. I thank you so much for talking about your beautiful work. It's funny you mentioned Dr. Alan Flake, because really early on in the talk, Arlene sent in a question that says she saw a talk by Dr. Flake at CHOP about the development of the artificial womb medical device. And if this is the one I'm thinking of, is that the one where they've got the lambs in like the bags?
Yeah, they are now starting along the route of moving that from the lamb. And Alan contacted me, and actually I'm going to serve on his advisory board to sort of make sure things are done well. My concern with these devices is that it's very invasive, right? So if you take a baby out of the womb, you put them in a bath of whatever, and you have to sedate, you have to do a variety of things that may in and of themselves at an early stage in development impact the fetus. So the verdict is still out. Whether it's useful to prolong gestation for a few weeks, but realize that when a baby is taken out of the mother's womb at that early point, she has a vertical C-section, and that precludes her for ever giving birth vaginally. There are many consequences to these actions, so they cannot be taken lightly. And I hope to serve as a strong advocate for the babies, as I hope to be.
That paper I've used that paper in team science workshops about if you're going to continue this work, who's on your team, basically. Exactly. And it's interesting. That's a great answer. Another question, this is from Karen Lobb, and it says, from a clinical perspective, how do you see your research changing clinical practices of treating preterm babies? Without adequate oxygen, very preterm babies won't survive, but so can you talk about how you weigh the consequences, both sort of the impact of your science in moving the field, and then what do you do with the individual baby kind of question?
So just like when we first started with talking about jaundice and hyperoxia and so forth, jaundice, too much bilirubin is bad for you. Too little is possibly impinging on your your antioxidant defenses. Too much oxygen is bad for you. Too little oxygen, of course, you couldn't survive. But what is being found is when people have tried to regulate oxygen to to keep it very modest for those young babies in the NICU to prevent some of the damage of too much oxygen, those babies actually had a higher chance of complications and death. So the challenge is finding that right, that balance. One of my slides usually is about the yin and the yang. You have to have a balance between good and bad. And sometimes, you can't go without oxygen. It's vital. It's a necessary evil. But how much? And how would this change practice? Well, if you better understood why and how metabolism is altered by oxygen, could you, by nutritional approaches, by other approaches, modify metabolism so that you could prevent some of the injury? If you knew that some of the cells in the lung, some particular cells, some niches of cells were predisposed to develop hyperproliferative compartments, could you find a way to target these cells to prevent that from happening? So this just gives you ideas on how would you utilize these things, changing metabolism, that's why we were going down the hemoxynase route, is we were thinking, well, could we use hemoxynase by delivering it or changing it or whatever? But it became more complicated as you thought about hemoxynase being important when it's in the nucleus versus in the cytoplasm. How do you do that in a clinical model? That is really difficult. So there's lots more to understand, and there are more techniques coming out that might allow us to be preventative by changing nutrition. My ideal goal is to change metabolism by changing nutrition. Something we don't learn enough about in medical school, and using that as a way to improve the outcomes. More to come. I hope that day will come.
And it really, it goes so well with your tree metaphor, right, Phyllis, that that it takes you down all these different branches. Once you start to discover, you uncover one thing and it's a whole other branch that's each gonna have little branches. The comments in the chat are all so positive, and I'm afraid we're out of time for today. I'm going to turn it back over to Carol to just say goodbye, and I think probably plug our next one of these Women in Science series. But this, again, what a bright spot, Phyllis, and a true pleasure to hear you talk about this work and your brilliant scientific career. And you've done so many things in so other realms, but this was so neat. I really loved hearing it. Thank you.
Thank you so
Carol.
much for having me.
Thank you, Dr. Dennery, again for an excellent presentation. I love the tree metaphor. And Katie, thank you so much for introducing us to Dr. Dennery and for your work on this project. And thank you all for attending today. I hope that you will join us for our next session on February 23rd at the same time. You can find this on Eventbrite. You can find it on our website. And I believe we're going to put the link to this in the chat function. If you don't see the link, then you can just go on Eventbrite and look up on the 23rd this session. So hope to see you there. It'll be a discussion about collaboration, since Drs. Rani and Nugent are collaborating on many projects together. So a little bit different focus on that one. So we look forward to seeing you all then. Thank you, Toonie. Thank you to everyone at the Venture Cafe. Thank you all again. We look forward to having you attend our February 23rd event. Please look on our website, www.ri-bio.org, for any other upcoming events. Please join us as a member or a sponsor for this series. We would love to have you be more involved. And I want to thank the committee again for all their great work on putting this together. So everyone be safe, be well, and happy 2021. See you all soon.
Thank you.
Thank you so much.
Amazing.
What a
Wonderful.
fantastic conversation. Thank you so much.